Bismillahirrahmanirrahim. This is the lecture series related to disease informatics. In the previous two lectures, we have discussed different types of data sets which are related to disease. Particularly, we talked about cancer and different types of genomics data sets. For instance, a whole genomic data set, whole exome data set, expression data set, that is RNA sequencing data set, and other types. And also we discussed the different genetic changes in that genomic or transcriptomic data sets, for instance mutations, copy number variations, different types of copy number variations, and then RNA expression. So today, now we will move towards the new way of looking into the genomics data set, particularly for the biologists and bioinformaticians. For instance, we have there are a lot of questions come in mind when we are talking about dealing with the data set. For example, if we need a data set that is related to cancer genome. Now, the first question that comes into the mind of any biologist or bioinformatician is that how to access that data set, right? For instance, first is what is the data set, right? Now that is the question first, or how to select. Now there are different types of data set as I discussed, like genomics, transcriptomics, right? So particularly in this tutorial we will focus on these two types. Now the next question that comes into the mind, that how to access this data, right? How to access genomics data set, that is DNA sequencing data set, and then transcriptomic data set, that is RNA sequencing data set. Now that is access, right? And then after accessing the information, for example, we now know that the data set is placed into this database. Then how to perform the analysis after accessing. Okay, before accessing, you can say how to download that information, then how to analyze that information, and in analysis how to perform different tests to find the right answer, right? So these are a few questions that directly comes into the mind of the researcher, be it a bioinformatician or biologist. So that is the actual problem. So to solve this problem, the different organizations and different institutions and different universities have come up with a lot of solutions and different data portals and databases where you can access all this information of genomics and transcriptomics. So here in this tutorial we will discuss one example. Like in the previous lecture we discussed about the TCGA, the Cancer Genome Atlas database, right? Now we will go into one portal from where you can find this data and access that data, and you can download that data, and then finally you can analyze and find the right answer. So here we will go to one portal, that is Cbioportal. Here you can see the name, cbioportal.org. In this portal you will find all the different data sets of different cancer types. C stands for cancer, and bio, bio, and then the portal, and different data types. For instance, in the left column you can see that there are different studies, for instance pan cancer studies, pediatric cancer studies, cell lines. If anybody is interested in cell lines, there are a lot of different cell lines, like more than 1,000 cell lines information available. Then adrenal gland, ampulla or water, any biliary tract, bone, bowel, breast cancer, cervix, eye, head and neck, kidney, lung, liver, whatever data set you want to analyze, which is available on this portal, you can access it very easily. There are a lot of different ways to analyze the data, but here I am showing you one of the easiest and fast methods where even biologists and bioinformaticians can access all the information and use it for their research. So let's see how the data looks like. For instance, now we want to, the question is, let's formulate a question, right? For example, the question is, how to find expression information of liver cancer dataset, right? So the first part of the tutorial is how to find the expression information of liver cancer dataset, and also the information of those sorry, you can erase it, information of those patients as well. For example, we want to see the expression information and the information of the patients, and we can correlate that with the expression. What I mean, I'll show you here. For example, now let's go and find the liver datasets. Here you can see on the left side there is a liver option. When you click that liver option, there are multiple datasets available on in the other side of in the middle of the portal. You can see here. For example, here one is hepatocellular carcinoma. Usually liver cancer, the primary liver cancer, most of the liver cancers are hepatocellular. So hepatocellular adenoma, you know the differences. This is beyond the scope of the study. We'll just focus to find the data. Then hepatocellular carcinoma, you can see here that hepatocellular. Then again, there's the name MSK. These are the different organizations which have submitted. For example, INSERM is a French-based organization. They have submitted the hepatocellular adenoma data in 2014, and that is published in Cancer Cell. This is one journal. Then hepatocellular carcinoma in serum, that is published in Nature Genetics in 2015. That is also available here. And there are other list of data sets. For example, here you can see TCGA. So this is the one data set we have talked about, the Cancer Genome Atlas. All these different tissue types, almost all of those contain TCGA datasets. For example, now we want to see that how the TCGA dataset looks like, right? Then let's click the dataset, or first we will see that there are different options. For example, here you can see TCGA liver hepatocellular carcinoma. Raw data is available at NCI, National Cancer Institute, and the source mutation data from GDAC Firehouse, and that is organized in a user-friendly way here. Then there is you can see there is a book type option. It's not available here. Book mainly refers to the PubMed. PubMed we have discussed about. It is available on NCBI, and all the literature related to all these data sets are available on PubMed. So if you want to access the published data, you can directly go to that link and you can download that information. Then this is the pie chart. You can see here is actually the information that is linked with this liver hepatocellular carcinoma TCGA Firehouse. So here you can see that in the first cancer type detail information, you can see hepatocellular carcinoma of 429 patients is available, and then there is a hepatocellular plus and fibromelar, different types. So in total there is a data set of 442 samples. Then here you can see molecular profiles. We have talked about methylation profile, then mRNA expression, that is RNA sequencing. mRNA expression z-score is actually calculated from this RNA sequencing, and this is a normalized expression. So we can use, this is also beyond of the study, we can use any of these two data sets. Then mutation information, that is extracted from whole genome or whole exome data set, depending on the type they have selected in this study. Then putative copy number alterations. These two are copy number alterations. So copy number alterations we've discussed about amplifications and deletion. Then this is the next, you can see overall survival information, disease free. We will discuss this in detail later, but if you want to go to the if you want to know that how to analyze this overall survival information, you can see one tutorial that is in the section of SPSS analysis. Here we analyze overall survival and we can get this output. So now first we have to look for the data. Then all these data sets are organized in different graphs and tables. This is the normalized data. But now the question is that how to find the expression information, right? Here you can see that this is the whole information. For example, now if we want to download all the information related to this data set, we can just click the download clinical data for the selected cases or the all cases, right? For instance, here we selected 429 hepatocellular carcinoma and we can download, right, from here. When you click the download, it will download the clinical information of this hepatocellular carcinoma patients. And yes, okay. And if we want to, here you can see that is all the clinical information. And now if if you want to look into the information, you can just go and see how it is organized. Here you can open it in any Word file. Here you can see that in the first column that is the study ID, that is LIHC. Then this is the patient ID, and then patient ID, and then all the information of different clinical features. We will discuss these in detail if necessary. Now this is the clinical information. That is actually the same information here, which is mentioned here, information of the patients, right? So this is the information of the patient, each patient ID, and then the respective information that what type of cancer it is, that is hepatocellular carcinoma, which clinical stage, disease stage it is, whether the data is metastasized, what type, whether disease recurred, or what type of population from this data is selected, right? Now this is the clinical information of each patient, and now we want to download the whole expression and copy number variation, all the information. You can just click at this and you can see that download all clinical and genomic data of this study. This will take quite a bit time. For example, this, hey, I just clicked it and you can see that this is of 101 MB file, right? So let it get downloaded and then we will discuss. Okay, when this will end, we can just unzip this file and we can extract the information of expression. One more thing is that, for instance, we want to download some specific. Now I will explain that how all the information of the expression can be extracted from here. In the meanwhile, just not to waste your time, we can put up another question here. You can see question number 2, that can be how to extract expression of set of genes. For example, we don't want to download all the expression data set, and we just want to focus on our gene set. For instance, we want to download information of, let's suppose, 5 genes. So again, we will go to liver cancer. Here you can see just clicking the liver cancer, and then here we will click the cancer. For example, here we have discussed about Firehouse Legacy TCG dataset. We will just click it, and now here you can see query by gene information. Just click it, and after clicking, you can see that there are different types of data profiles associated with this dataset. One is mutation, that is whole genome genomic sequencing data. Then copy number variation, that can also be whole genomic or SNP array data. Then this is the expression information. Right now, our question is related to expression. So when we click the expression information, here you can see that the information of mRNA expression is available for 373 patients. Now we want to set some genes here so that we can look for those. For instance, we want to look for GLY1 gene, GLY2 gene, GLY3 gene, right? Sonic hedgehog gene and Indian hedgehog gene. So these are 5 genes as we discussed. Now after writing all these genes, you can just submit the query and it will process the information, and that is okay. Now this is opened here in a graphical way, this is called oncoprint. Here you can see that GLY1, out of all the 373 patients, this each bar represents one patient, and out of 373 patients, GLY1 is overexpressed in, or you can say mRNA is high in 5% of the cases. This red represents mRNA expression high, and the gray represents that there is no expression alteration, that is the normal expression of GLY1 in the rest of the 95% patients. Then there is a GLY2 0.3%, very less, then GLY3 4%, as Sonic hedgehog 6%, and Indian hedgehog 5%. Now this is the overall expression information of these 5 genes, but this is graphical, and now we want to look further, that we want to download the information now, and for that we have to go into the option of download, and from download here you can see that the mRNA expression of the gene set is available here. For instance, if we click this tab, delimited format or transposed matrix, whatever you want to download, if you downloaded it, it will be saved here. And here you can see now that all the 373 patients, these are 374 lines because the first line contains the header file. So the 373 patients, the expression information of each gene, that this is expressed at this level, and this, you can download all the information like this, right? Now, okay, so this, I think this is very easy to follow, to download the information. Now here you can see that this one is also downloaded. Let's see that if we want to look for all the expression data, for instance, there are, you know that there are almost 22,000 protein coding genes, right? Now we want to see that how the information is organized. That is very similar to this fashion. For example, here you can see that there are only 5 genes, but in that data set there will be 22,000 or any all the information that is available for this data can be placed in front of this, is placed in this file. So let's see why it's not done yet. Okay, wait, let's see. Okay, so waiting is always very difficult. Okay, yeah, now it's done. Now this is all the information that is available. So for this tutorial we are just focusing for the expression information. So we can just find the expression information here. You can see that this is the methylation information, this is the RNA sequencing information, and this is the Z-score information that is also available on the we already downloaded for 5 genes. So we can open it straight away. Yes, now you can see that now this is this is different from here in a way that here all the samples are arranged in columns and genes are arranged in rows, right? But this file is all the genes are arranged in rows, in columns, and all the samples are arranged in rows. So if you want to make it same, you can just go there and you can just download this transposed matrix, and both will be the same. Just to make you don't make you panic. Let's see, right, like this. Now all the samples are arranged in columns and genes are arranged in rows. Okay, so here you can see that all the information of all the genes available for this, for example, here this information is available for 20,532 genes is available, and you can extract the information. For instance, if we want to look for GLY1, here you can see, you can just search it and you can find the GLY1 information is also available here, GLY1 to GLY3, and also if you want to search SHH or IHH, you can find here. So in this way you can download the expression information very easily, and you can just select that information and copy in another file if you want to analyze only these 3, 4, 5 genes. And if, or depending on your question, if you want to go for all the data set, that's up to you. Now this is, now the next question should be, now we have we know how to download the expression information, how to download the clinical information, and now the next question can be, if I write it here, where is the, okay, so next question will be, I'm just writing all these questions for your ease and also for mine, so that I have to give you exercise later on, and that will be, in this way you can also learn a lot. That is how to analyze or correlate gene variation with clinical information. To make this question more simpler, it means that now obviously the gene variation is actually causing this cancer, for example, right? Now which gene expression or variation is actually associated with the clinical information? For that we need two things. One is the clinical information and the other is the expression information, right? So for using these two informations, this expression information and to make it look good, let's go back to the, yes, this file. For example, now we want to see that how GLY1 gene expression is showing variation with the clinical data set. So you can just compare this clinical patient information with this data set and make one file which contains both GLY1 and clinical information, so that you can compare that whether the GLY1 expression has anything to do with the clinical information of this hepatocellular carcinoma patients. So I think now we are going into the further deep details about comparison and analysis. So first of all, I think the purpose of this tutorial is almost done and now you know that how to find the expression information of liver cancer dataset. You can change this liver cancer to any breast cancer, cervical cancer, any other cancer, and you just search and just download using click, and or you want to download all the dataset, then you can go on that chart and click and then go and download the whole dataset from that this portal, and then information of the patient can be downloaded from that clinical information data set which I showed you previously, and also we have also discussed that how to extract the expression of set of genes we want to analyze. Then here in this part needs further discussion, that how to analyze or correlate gene variation with clinical information. So now we have the gene expression information and now we have the clinical information. So for the next step, what should be the next step? In the next step, we have to make another file which contains, for example, the 5 gene information with the clinical information, and then perform different statistical analysis to find any association between the genes and cancer. For that, I will in the next tutorial I will discuss that in further detail, to that how to analyze and compare these two information. However, in my previous lectures on statistical analysis, you can find these different ways to analyze this information. But to make it more simpler and clear, I will try to make another tutorial so that we can link this third question to the next part of the clinical and gene expression analysis. I hope this is clear now, that how to what type of data set we wanted. We wanted transcriptomic data set. How to so if you write it here, what type data set we wanted here, that was transcriptomic data set, and then how to access it. For that we went to Cbioportal, right? And then how to download, we just clicked the sorry, it's too dirty. I think I should show it here. Click download after selecting the study and also the clinical information, and then analyze. For analyze and to find right answer, we need the expression data which we have already downloaded, and now we have to perform different tests after making the file which contains both the expression information and the clinical information. So for this part and for the file part, we will come later and discuss that in further detail. Thank you very much. I think that should be enough for now, and I hope you are following my previous 2 lectures and now to this lecture. So how to link that information with the previous lectures? Obviously, previously we discussed in general about the different data set types and different types of sequencing methods. Now we have selected one data set where we can find that genomic variation information and the sequencing data sets to come to make it more clear. Thank you. I hope to see you in the next class. Assalamu alaikum.